Hi, I'm Maddie.
I'm Mia.
And I'm Neha.
And so our project is about the gut microbiome's effect on autism.
So, autism is a mental disorder and is identified by a spectrum of difficulties, including social and communication difficulties. It can be developed through genetic predisposition or through environmental aspects that affect the gut microbiome. An unhealthy gut microbiome affects the development of autism by causing inflammation in the body that travels to the brain and can affect the development of autism. Lisa Aziz-Zadeh, along with scientists from UCLA, were some of the first to research the connection between the gut microbiome and the brain. Since the discovery of this connection, some treatments have been developed, such as a fecal transplant, where a healthy fecal sample is transplanted into the digestive tract of a person with autism in hopes of creating a healthier gut microbiome. This transplant takes about 2 years to show results.
The gut microbiome is a group of millions of microorganisms that reside in the stomach and the intestines. It is a rapidly changing environment of bacteria that is very vital in bodily health and regulation of disease. Though there are many trillions of microorganisms in and on humans as a whole, the gut microbiome in particular directly correlates to the different functions of the immune system. A balanced system ranges from controlling digestion all the way to managing allergic reactions, while an unbalanced system can cause problems including obesity, diabetes, and more recently, autism. Through recent discovery, it has been proven that kids with ASD have different gut compositions than those without. A recent study this year done by UCLA and USC scientists proves that gut bacteria activates the central nervous system to send information to and from the brain. Additionally, knowing that the gut is largely affected in diet, new studies have emerged to restrict what children diagnosed with ASD digest. Yet these studies prove to be inconclusive because not all severities of autism can be lessened by a change in diet. However, by analyzing the fecal transplant bacteria properties held in stool samples, scientists were able to come up with a connection between said properties to known behaviors in children diagnosed with autism.
During the 1800s, a scientist from Boston, Massachusetts began recognizing autism as a mental disorder. Scientists were interested in how people with autism were challenged in some areas, but at the same time extremely talented. At this time, scientists were not 100% sure of what autism was. The word hadn't even been invented yet. This new discovery sparked scientists and researchers' interest in studying this newfound disorder. During these early stages, scientists and researchers diagnosed autism by watching and analyzing the behavioral patterns of an individual. It was not yet known that the gut microbiome had anything to do with autism. Scientists continue to study autism to this day, but at first had very limited knowledge on the subject.
The discovery we found was that the microbiota transfer therapy, commonly known as a fecal transplant, were widely successful between those who have a purely healthy gut microbiome to those children who possess an unbalanced microbiome due to being diagnosed with autism. A recent 2019 study by Arizona State University concluded that 50% of children who were diagnosed with autism, that autism symptoms reduced nearly 50% 2 years after the fecal transplant. The way it works is relatively simple. As the gut microbiome of a healthy child has stronger and more valuable bacteria, if you take the specimen from the feces and transplant it into the digestive tract of a child diagnosed with ASD, the hope is that the bacteria would create a healthier environment in the gut so that they can balance their microbiome over time, which proved to be very successful.
So, autism can either be caused by a genetic predisposition or it can develop through environmental influences that affect the gut microbiome. An imbalance in the gut microbiome can cause inflammation in the body, which can travel to the brain and may result in the development of autism. Lisa Aziz-Zadeh from the USC Division of Occupational Science and Occupational Therapy stated, "If you ask the parent of a child who has autism, there's a good chance you will hear stories of tummy troubles like constipation, cramping, and pain. In this quote, Azizadeh highlights the external signs of the internal imbalance in the gut microbiome. The gut microbiome is responsible for many important bodily functions such as immune system and metabolic functions, regulating disease, and vitamin production. A fecal transplant is a newly developed treatment that has shown promising results. In a study by ASU, as was just stated, they found that 2 years after the transplant, 50% of the patients decreased their symptoms by nearly 50%. This discovery is just one step further into the future of treatments for autism.
So our ideas for the future is we will be using an optical light microscope to study the detailed structure of the bacteria in a healthy gut microbe versus an unhealthy gut microbiome. Our plan is to improve the mechanisms of the fecal transplant by minimizing the amount of time that it takes to reduce the severity of autism symptoms. As previously mentioned, the microbiota transfer therapy takes about 2 years to see its full effect on ASD symptoms. For people that are higher up on the spectrum, this is 2 years of life that they have to live before change starts to occur. Our action plan is to infuse the species of the feces before this transplant with naturally good bacteria such as raw probiotic strains. Probiotics are live bacteria and yeast that are generally very good for you, and when ingested, they can take residence in the gut and naturally improve your health. By infusing the species with probiotic strains, the goal is to increase the rate of growth for the amount of healthy bacteria to keep the microbiome sustained. Of course, further research has to be done to see if the feces would react accordingly with the probiotic strains.
Contributions. One major contribution was the discovery of the fecal transplant. Stated by the medical press, fecal transplants help to substantially improve behavior and gastrointestinal health in children with neurodevelopmental disorder. These transplants are not used from pure fecal matter, but instead purified matter that has been extracted from feces. Fecal transplants rebalance the intestine, which directly influences the brain. Introducing this new bacteria diversifies the bacterial community, also stated by the medical press. After the transplant, and now a new abundance of beneficial bacterial species was formed. The fecal transplant has majorly contributed to helping kids with brain disorders like autism.
And these are our references.
Please remember to repeat the question. Okay, so there we go. Thanks. Okay, now you don't have to repeat it because they can hear me. Okay, so I wanted to clarify what exactly you would be looking at with the microscopy technique that you mentioned. So you said optical microscope, and you mentioned wanting to look at the detailed structure of the bacteria. So what structural features are you talking about specifically, and how big are those features? So I guess that's connected to saying, is optical microscopy actually the technique that you want to use to see what it is you're looking for?
So I think what we would do is we would inject GFP, green fluorescence protein, into a stool sample with an active gut microbe in a healthy sample. And with the probiotic strains that we would inject into them, we would see if the probiotic strains would help increase the amount of bacteria that is created when we put in the fecal transplant into the digestive tract of a kid with ASD.
And also I wanted to add to that, so the reason that we chose light microscope is because we wanted, if we were to use the electron microscope, we wouldn't be able to use a live sample, and all the microorganisms, even though they're tiny, they're really tiny inside the microbiome, and we wouldn't be able to use a live sample of that and see everything that's happening inside of there. So that's why we chose the optical microscopy technique.
I truly find that fascinating that you've taken a really clinical problem and are trying to explore why, what the cause of it is. I mean, fecal transplant is a clinical issue. Autism is a spectrum, right? So it's a spectrum of disease. Have you read anything about the fact that perhaps there'll be different gut flora or microbiome changes depending on what spectrum of the autistic spectrum one would be on? So you're taking a clinical problem and you're trying to find a microbiological cause, which I think is fascinating, actually.
I can answer this one. So wait, I don't need to repeat it, right? Yeah. Okay. So yes, everybody's microbiome is going to be different. What the fecal transplant is trying to accomplish is just introducing more and like healthier microbiome into someone with autism who may have an imbalance in their microbiome. So everybody's microbiome is different, but the fecal transplant can help to fill in those imbalances.
I can talk loud. So, in your proposal to utilize optical techniques, do you see that what you learned and saw this week is actually capable of pulling off the measurement that you want? And what I'm getting at is that the optical microscope can certainly visualize bacterial cells. It can image, over large areas, living organisms. It can measure dynamic processes. But did the microscopes that you saw today or this week indicate to you that we're ready to try to do this measurement inside of a live human, for example? Because we talked a lot about how you actually gain access to your sample, right? And the reality at some level is that the gut microbiome is inside of my intestinal tract. How do you get the measurement that you want to do to operate in that environment? Or do you plan to prepare your sample in some other way? If so, what are the challenges with doing that?
I can answer this question. So we read and researched that a lot of studies have been done on mice, because mice and humans have a similar digestive tract and gut microbe that we could study potentially for the bacteria. So additionally, like for current procedures, I guess we don't exactly know how the humans would react if we were to look at their bacteria just straight from their gut microbe. That's why a lot of studies have been done on other animals, such as like mice, that we said, if that answers your question.
Yeah, I guess I was asking, are those studies that you looked into done in live mice inside of their intestinal tract, or are they trying to basically extract, purify, and recapitulate the microbiome in a petri dish, for example, right? Because there's a big difference between what you can do in a dish by just mixing together a bunch of bacteria versus what actually happens inside of your body, because it's not just what it's made of, it's also where it is. The spatial distribution matters, right? So I guess I'm just wondering, in your research, did you see that these technologies currently, and it's okay if they're not, right? Because part of this was, what do we need to do, right, to identify new tools and technologies to enable us to do this type of stuff?
I think currently, I'm not sure if the mice were alive. I think they were just taking out samples of the gut microbe so that we could see the bacteria and see how it, enhances after the fecal transplant is put in. So I'm not sure if there's enough research out there yet to do live gut microbe screenings, but I think that's definitely something that could be looked into.
So I could maybe add a little bit to this. One of the current technology that has been developed is actually, as we touch upon very, very lightly, I don't know if you saw that later on by looking at the literature, is to miniaturize microscopes, right? So one of the very important effort here is to take the microscope and bring it to the tip of an endoscope, for example. So you can think about using an endoscope coupled with a microscope to actually look at the microbiome inside a live mice or a live human.
Yeah.
And this is actually probably going to be done very soon.
Yeah, the studies that we saw were very recent. Most of the studies were in April and May of 2019, so they're very new in coming up with their research, so there's definitely going to be more out there soon.
Thank you.